Good evening, everyone. With great honor and pleasure, I, Roshan Zehra from the Department of Pharmacy, welcome you all to the occasion of our college alumni connect activity. This day is very important as it provides an opportunity for everyone to meet each other. It also provides a chance for every graduate from the previous years to meet their former teachers and the other staff members. Our college has established a tradition to stay with their contacts. Well, continuing the tradition, our college has organized this e-symposium and I wanted to welcome Dr. Pratik Pathak, Senior Research Scientist, South Ural State University, Russia, who will guide our students for the topic drug delivery research. Now, without further ado, we are going to start this session. Please welcome Pratik. Please continue, Pratik.
Hello, everyone. As Professor already told me about that, my Pratik. I did my graduation from IIMT College of Pharmacy in 2010. I was the first batch student. So probably you don't know me. And today I'm going to present our drug discovery topic, that how any research group or any scientist are going to design a drug and deliver in a effective way. So here I'm going to present one of my research paper, that is designed to in vivo studies of hybrid quinozoline 135 triazine derivatives as an epidermal growth factor receptor. This is EGFR receptor. So first of all, what is EGFR? Means epidermal growth factor receptor. Because now we are going to discuss the inhibitors of this. So now I'm going to give a small discussion about EGFR. EGFR is a transmembrane protein. Means transmembrane, the means is that the protein which is available on the surface of any cells. So it is a outbounded surface protein which belongs from a tyrosine kinase groups. It participates in the transportation, transcription of the regulatory molecules, and due to the result of this regulatory molecules transcription, cell proliferation, differentiation, apoptosis, and angiogenesis take place. So this is the normal physiology of any EGFR protein. But in the case of tumor, this EGFR get overexpressed or get altered or changed. So because of this changing, the process of angiogenesis is increased, and due to the result of this, lung, breast, colon, and bladder type of cancer generate. So we can say that if EGFR receptor change or alter his normal physiology, it leads to the formation of cancer. So this EGFR identified 30-40 years ago in 1970s time. So from 1970 to till date, there are several drugs which are introduced to inhibit or to target EGFR, endothelial growth factor receptor cycle or pathway. So these drugs basically bind with intracellular domain, or it can bind reversibly or irreversibly. So reversibly, as name suggested, means if the drug bind with this type of receptor, it after some time it get out from that pocket, and physiology of that particular cell get normal. Irreversible inhibitors generally bind with the cysteine protein, or allosteric binding sites of that particular protein. On the basis of that, the complete killing or cytotoxicity of the cancer cell take place. So there are several examples of EGFR class of the anti-cancer medicine. The first one is gefitinib, second one is erlotinib, third one is vendatinib, lapatinib, osmetinib. There are several types of the drug. All the students must be aware about all these drugs, because this is as the pharmacy second year students or third year students, they are studying pharmacology or medicinal chemistry. So whenever you are going to study about anti-neoplastic medicine, you will get all these type of names. These are first generation, second generation. On the basis of their fragment and discovery, they are classified. So these are the examples of EGFR receptors. Let's go to next. Now here our study is going to start. In our study, we selected two different skeletons. First skeleton is quinozoline, and second skeleton is triazine. So as again, I'm going to my previous slide. You can see in all the structures, any guys who have interest on medicinal chemistry, they can see from all the structures, all the maximum these compounds, maximum this erlotinib, gefatinib, lapatinib types of drugs contains quinazoline rings, which I can show here if it is available. So this all the second to bicyclic ring, where one is benzene, second is pyrimidine ring. These are this is quinazoline ring. So from all the classification, we can understand that quinazoline is very important key for the anti-EGFR activities. So in our research, we selected quinazoline first ring, and second ring was triazine. Why we selected triazine? Because triazine is a compound which have symmetry, plane of symmetry, as we can see from the structure of triazine, where second structure on the uh, right side, we can see that there is 3 nitrogen atom present in the benzene ring, and all 3 atom is arranged in a such manner, first one nitrogen, then then one carbon, then one nitrogen, then one carbon, then one nitrogen, then one carbon. So this is plane of symmetry. So that's why this is 1 3 5 s triazine, means first, third, and fifth position of nitrogen atom, and which contains a symmetry. That's why it is s triazine derivative. So uh, so we decided to select these two compound, and we went to the research, and we found from the literature that from our previous research and some other scientists research that chloro, bromo, and nitro groups have important role when we add in the structure. It increase the biological activity. Secondly, if we add any aliphatic group between quinazoline and triazine ring, the activity will decrease. But if we add uh, any other ring, it can be enhanced or it can be changed the activity. So in this paper, we measured this. What is the role of aliphatic or aromatic chain? And third is the adding the bulky group, which is generally primary and secondary amine, on the third position on carbon. You can see from the structure, the R is there. What is the effect of this R group if we add some bulky group? So this is the complete hybrid structure we decided to synthesize. That this is the normal synthetic process. The first step was the conversion of was the conversion of dibromoethanolic acid into the quinoxazoline ring. So this is the normal cyclic formation, cyclization reaction, where carboxylic acid converted into the pentacyclic ring. The yield of the product was 84%, which was quite good in the term of medicinal chemistry. Second reaction, which is termed as B, it contains the addition of the aromatic group on the by the replacement of oxygen atom from the bicyclic aromatic ring. So we get our third product. At the same time, this reaction carried out in the two parallel direction. First direction was formation of from 1 to 2, 2 to 3, and another direction was from formation of 4 to 5, 5 to 6 molecules. So you can see fourth compound is cyanuric chloride. We made mono by using this cyanuric chloride. We made mono substituted cyanuric chloride, and then again in next step, which was D, we clubbed two different amine groups, and we found two di-substituted cyanuric chloride. In the final step E, we combined compound 3 and compound 6 and find our final molecule, which was 7. And there was a series of number of molecules, so 7A to 7N means 7A, 7B, 7,6, like this. We synthesize different molecules, and you can see A, B, C, D, E, F from the right side of the corner. There are different type of substitution which we used. This is the normal synthetic method for synthesizing this drug. Further, we move on in vitro results, in vitro activity, because in medicinal chemistry, whenever we are synthesizing any medicine, we have to check it is effective or not. So for cancer, it is the primary testing to perform in vitro MTTSA. So for MTTSA, we selected 4 different cancerous cell line and 2 normal cell lines. So first cancer cell line was the breast cancer cell line, that was MCF7. It is the term which given by the scientific community to a specific type of cancer cell line. So MCF7, HeLa, this is for cervical cancer, HEPG2, that is for hepatocellular cancer, HL60, which is for leukemia, HFF, which was normal foreskin fibroblast, MCF12, which is normal epithelial cell. So there one question can be arise, why we take 4 cancer cell line and 2 normal cell line. So this is in biological activity evaluation, our main any scientist have main target that drug should affect the cancer cell but should not affect the normal cell, because if drug affect the normal cell too, then the drug will be very much more toxic and you cannot use as a medicine. That's why for checking the selectivity of our drug we use 2 normal cell line to evaluate that. And after that we performed EGFR inhibitory activity, because we want to target EGFR receptors. So we need to check that drug is effective against this particular enzyme or not. So we checked against EGFR enzyme, and further we performed anti-ovo-angiogenic activity against MCF induced chick embryo. So in this part, we took a chick embryo, we introduced MCF, means breast cancer, into the embryo, and then we checked the activity. So from the result, you can find that our 4 drugs, that was 7C, 7D, 7E, and 7J, have quite impressive biological activity against during the MTT in vitro assay and EGFR inhibition. And we found that our drug 7E have, in all of the series that come, the drug 7E have most prominent activity which are near to similar standard drug erlotinib. Erlotinib is a marketed drug which is already using. It is a standard anti EGFR inhibitors which is widely used worldwide. So our drug is effective. How we can say that anything is effective? So if we check our things to compare with another things, so that's why we check our drug with some standard drug, and we find that in all of the series, the compound 7E is most effective. Further, so this was the experimental aspect. Now we wanted to identify that how drug act in the biological system, how drug entered in the, and how it's bind with the protein. So for that we perform in silico studies. In silico studies contains docking and molecular dynamics. In docking we performed docking study. Means docking is a tool where we fix the protein structure and try to introduce our drug. If during the microbiology session every student should every student must be studied that drug and protein and drug are like key and chain, key and lock. So here you can, everybody can understand, like protein is a lock and a drug is a key. So we checked that what the key we made, it was exactly going to fit with that drug, that protein, that lock or not. So by using the docking technique, we can identify this thing, and we can identify and we can find that how much drug is effective, how much strong bonding happens between the drug and receptor. The bonding which will be strong, activity will be strong. So here we compare and we find that our compound 7C, 7D, 7E, and 7J have higher value that erlotinib, which is already standard, and and also higher than second standard medicine lapatinib. So on the basis of that, we can say that the drug which we synthesize have maximum chance to bind with the protein, EGFR protein, and can show the biological stable structure. Now further, we move to the molecular dynamics technique. So molecular, there in silico drug designing, there are 2 terms which is widely used. First is molecular docking, second is molecular dynamics. In docking state, we assume that protein is rigid and our drug is protein is rigid and our drug can be flexible. But in molecular dynamics, it is a like that system which is like biological system, because in docking only 2 factors, protein structure and drug structure only matters. But here in dynamics we we introduce all the parameters, all the biological parameters which is in a living cell. We introduce here in a simulation, like addition of water, management of the pH, because when in a biological system or any life, any cells or any body, any organism, all the organ have their physiological balance, their pH, their basicity, their how much they have pressure, uh, internal organ pressure, what is the mechanism, viscosity, velocity, everything occupy within the cell. So here we checked, we applied this all the parameters and checked that how much our drug is effective or how much it is inside the protein pocket. Because if drug is inside the protein pocket, it can be work. But if drug get out, it will not going to work. So here we checked and we find that our all these 4, 7C, 7D, 7E, and 7J have highly effective property to bind with the protein and to form the intermolecular hydrogen bonds. So this here I want to focus, tell one more thing, that hydrogen bond basically in between the drug and the receptor, for activity, hydrogen bond have most significant role, because hydrogen bond, it is not like that drug go bind and it cannot be removed. Drug should be removed, otherwise your physiology, body physiology will change. Because every drug that's why we are studying half life, because after whatever the hours, like 6 or 10 or 12 hours sometimes, so after that hours this bonding ruptured, and after that drug get eliminated from the body. So because our biological system is like that, any drug cannot be bind for whole of the life. It cannot be possible you eat the medicine and it will be for 100 years. No, because it will change your physiology. If anybody make a medicine which always get to the bind, will not remove, then your that particular receptor get destroyed, because this type of interaction give the pressure to our physiology. So our physiology change, so nature of the person, nature of the organ get changed. So it is important to drug should have some half life. It should be delayed or it should be fast, but it should be. So that's why we check there. Now, after encouraged by the result, we went to the in vivo activity, that was anti-breast cancer screening. So, in vivo assay is the assay where we use the live animal, for evaluating our primary research, in vitro assay, our research. So here we took female rats, grossly sparse rat, and this is a specific type of rat which are using for the evaluation of biological activity. Pharmacology branch students must aware about this. In this, so we take this and we introduce our drug and check the body weight. First we introduce the cancer in the rats, then we introduce our drug, and we identify that how much our drug effect against tumor, it's decreasing the size of tumor or not, it decreasing the tumor volume, it increase the body weight. Because we know that if any patient have cancer or anything have any animal have cancer, the body weight decrease so rapidly. So if drug have effect, so body weight also improve. So we find that finally, on basis of all the absorption, all the parameters, we find that our drug increase the body weight, decrease the tumor incident, tumor percentage, and tumor volume. Apart from this, we also find that our drug act on enzymatic and non-enzymatic antioxidants, which is very important during cancer treatment, because all the enzymatic and non-enzymatic parameters, antioxidant parameters, that is CAT, GPX, GSH, SOD, there are several. So this alterly get rapidly changed. So drugs should be responsible to decrease this also. So our drug was decreasing this. So we find that all the value return to the normal range when we introduce our drug to the animal. And after that, we also find that our drug also effect on the thiobarbiturate acid reactive substance, TBRS, and other LOOH parameters. So the overall we'll find that the compound 7E was the most effective, and it have significant property to inhibit the particularly breast cancer. Sorry. We also check the enzyme, other biotransformation enzyme parameters, because this enzyme parameters is very important for the uh, excretion of drugs. So we identify, like, excretion means, if we go to the literature, we found that any drug which passes by 4 different steps, A, D, M, E. So it is biotransformation is a part of E, excretion. So the excretion contain two phases, that is phase 1 and phase 2. And all these phases depend upon several enzymes. Like phase 1 depend upon cytochrome 450, and phase 2 depend upon GST and GR. So we found that our compound decreased the normal value of cytochrome 450, and also increased the normal value of GR and GST. Apart from this, some studies elaborate that during the cancer, the lipid profile get changed so much, alterly significantly changed. So it is important to identify that of any drug which is going to test, if shows any effect on lipid profile or not. That's why we checked lipid profile too, and we found that our drug significantly decrease triglyceride, HDL and LDL in a dose dependent manner. TC and VLDL uh, maintain at the normal values. Finally, on conclusion, we can say that we successfully delivered 14 different quinozoline 135 triazine derivatives, and we found that their four compounds have significant in vitro anti-cancer activity, whereas compound 7E have 7E induces EGFR conformational changes, and we showed that compound 7E have stable property to bind with the protein. After that, from the in vitro assay, we find that all of the drug, our main compound 7E have significant property as anti-cancer medicine via inhibiting EGFR pathway. So this is the main structure which is drawn of 7E compound. This was the drug which was most effective during the, which was found most effective during the analysis. So from this, from this structure, we had a good opportunity to discover new anti-cancer medicine, and our team is continuously working on this. Let's hope we will get some successful result in future. This is my publication on this topic. The topic is hybrid quinozoline 135 triazine derivatives as EGFR receptor with anti-cancer potency. This publication I published in ChemMedChem. It is a quartile 1 publication, which is already available online. If any student want to read, they can access on the internet. Thank you very much.
Thank you so much, Dr. Pratik, for taking time out of your busy schedule to enlighten our students about the topic, the drug delivery research, and explaining the topic and about the EGFR inhibitors very well. And I also want to thank all the participants of the event for joining this. And now I would like to call Dr. Sudhir Kumar for giving the vote of thanks.
Well, thanks very much, madam. Well, very good to see you, Pratik, after such a long time.
Yes, sir, it's almost a decade.
So, well, I feel very happy that you have progressed a lot in your life, and definitely your education from IIMT given you a seed for further going. And we are very much delighted to see you here and coming among our students out on online platform. We'd like to see you in future here physically as well. And thanks very much on behalf of IIMT College of Pharmacy, Greater Noida. I express vote of thanks to you for being here and giving your time, valuable time out of your busy schedule. Well, thank you very much, Pratik. Thank you very much.
Thank you, sir. Have a good day.
Thank you.
So last but not the least, I would like to thank everyone who has participated in the event. Hope you all like the arrangement and wishing you a wonderful evening. Have a good time ahead.